want to understand how the 3D context modulates signaling.
Yeah.
And so there are different ways how this may happen. So for example, we work quite a lot with epithelial cells that are polarized cells, right? So they have a specific apical domain, a specific basolateral domain with different proteins. So how this polarization affects signaling, localization of receptors, secretion of ligands, and so on and so forth.
Yeah, and I guess especially in the context of epithelial, and I'm thinking about the epiblast structure. You also have more how to say that, like topological effects. For example, the lumen is actually some part of the embryo which the content can be controlled. There can be secretion in it. There can be very localized signaling. So the paracrine autocrine influence between cells might be very different in 2D and 3D.
Yeah, I mean, you are just describing one of our projects.
Okay.
So exactly, we really want to understand what is the function of the lumen, absolutely. Yeah. And actually, if you think about, so let's think about the embryo for a second. You have those epiblast cells, like you said, they are all facing this central lumen. It's a very small cavity. Whereas if you think about just standard 2D culture, all the autocrine factors that the cells are secreting are going to be diluted in this huge amount of media. And actually, we have calculated that there is a 2,500-fold difference in the concentration of autocrine factors when you compare the lumen to a standard 2D culture. So definitely that is going to have an influence in the cell. But how is that influencing cell behavior? And also, what are those factors, those autocrine factors that are apically secreted? These are questions that we are addressing right now in my lab.